John Beckmann, Professor John Beckmann, Dr. John Beckmann, the father of Cid, the transgenic mother of sin, your hostess with the mostess. Get a green screen in the back. Hopefully it doesn't turn black. I drink my coffee black. I have my brain tied behind my back just to make it fair. What's that comment in the chat? What's my cup say? It says, Wolbachie, shut the f up and give them species names. A cup from Jogel. All right, part 2 of the structure paper. Let's finish the discussion. Where did we leave off? I think we leave off on figure 2. Hello? I'm filming right now. Is this an emergency?
No, I was worried. I've been calling you for over an hour because I thought that you were still home and then you didn't answer and I was worried. I thought you might be in an accident. Sorry to care so much.
I rode the motorcycle.
I know. That's why I was worried when I got home after I hadn't heard from you. It's okay. I'm glad you're okay.
All right, here's figure 2. Let's dissect figure 2. So A is kind of attacking 2x1 via the yeast assays. So again, 2x1 hypothesis is this idea that you need 2 to induce CI. We've known for a long time in yeast that all you need to get a yeast phenotype is just the B, and the A's never do anything, never do anything in yeast except rescue the phenotype of B. So in the yeast data, rescue has always worked. Rescue has always been a perfect toxin antidote model in the yeast. And here they're kind of like attacking, chipping away at the 2x1. So here's a positive toxicity control, Cin B, expressed with also an empty vector, kills the yeast cells. Here is Cin B with its cognate antidote. You get full rescue equivalent to the wild type. Here is Cin B with Cin A that cannot bind to Cin B. So the 1-3, that's the mutant that cannot bind to Cin B. So without the ability of B to bind A, you still get complete toxicity, complete death, okay? Here's a reciprocal, Cin B that can't bind Cin A, okay? By itself, kills the yeast cells. That's chipping away at the 2x1. Here's the Cin B that cannot bind Cin A. This would have been nice to have be a little bit more perfect. There's definitely like a little bit of interaction it looks like happening. It would have been nice if this was just completely blank, unless I'm misinterpreting the data. But it's definitely not like, it's not giving its full activity. Okay, and this is really interesting. This is where they're both inverted. So this is the Cin B where all those charged residues at 1,3 are inverted. So let's call this the negation. And here is Cin A where all the interface residues at 1-3 are inverted. So we'll call this the negation. So if you negate both, you take the opposites at both those interfaces, it reconstructs the ability for those proteins to now match and bind each other. So you can literally just switch here and switch there, and then it reassembles the ability to completely bind because you get a complete perfect rescue. So this is a really nice, interesting experiment where they're doing the double inversion, and the double inverted now binds perfectly and rescues perfectly. Okay, and then in C, they're doing the same experiments, but with transgenic Drosophila. So here's the negative control hatch rate, about 95, something like that. It's about right. Here is the CI control, getting very nice, strong CI here. Here's the rescue control. This is interesting. They're getting a very strong rescue effect, okay? So essentially a mean of almost 0 to like up here. But it's interesting. They're seeing a bimodal distribution, which I have seen before. This is interesting. This is interesting to me. You say you want me to explain what I think the bimodal distribution might be. So again, like I feel like rescue is very finicky. Rescue of CI can be finicky in a sense that I think the concentration of these proteins are important. So if you don't get the stoichiometry right, you can create scenarios where I think it's impossible to rescue. Like I think again, like what I tell my students is, I think that CI is like you're bringing these embryos to like a point where like they die, and at some point you kill them too efficiently. So like if you imagine yourself like if you stab somebody with a knife, they can probably go to the doctor nowadays and the surgeon can fix up their problem, they can be rescued. But if you stab them 50 times, like you can't rescue, like that cannot be rescued. And I think that's what's happening with CI. Like I think that these toxins are causing kind of like a gradient of toxicity. And if you go too far, you can't rescue. So my suspicion of what these bimodal rescue distributions are is I think that when you're comparing embryo to embryo, they might be inheriting different rates of essentially like the zygotes might be encountering different concentrations of the toxin. And in certain situations, they might not be able to rescue. And because this is a transgenic system, it's not evolved to be perfect, right? Like in the Wolbachia system, it's evolved to be a perfect system so that it works. And so you probably wouldn't see these bimodal distributions. And Wolbachia are living organisms. They can distribute themselves throughout the germline to get, if they want, I guess, or not if they want, but if they've evolved that preference to be more homogeneous, you might get more consistent phenotypes. So I think this is a relic of kind of like the transgenic systems when you see these bimodal rescue distributions. Okay, and here is induction of CI crossed to an antidote that cannot bind the Cin-B cognate partner, and you don't get a rescue effect. So you cannot get rescue if you do not have a binding interface. So the key conclusion here is that rescue is essentially equivalent to binding. Binding is necessary for rescue. And this proves the TA model, which all along, as I discussed in the previous video, has been that rescue requires binding. And here's the fly data to prove it. Very nice. Very nice. So here, this is interesting. So now they switch to Cid-B, analysis between Cid-B and Cid-A. And this time what they're doing is they're making an alignment between WPIP and WMEL, expecting that those cognate partners would be bidirectionally incompatible. And they're switching the residues in PIP to resemble the residues at those positions in WMEL. So almost like mimicking a bidirectional incompatibility in nature. So that's these data and the pull down data, where the crossed ones are the flipped residues switched to the WMEL. And here you see cannot rescue, but cognate partner does rescue. And here's the toxicity control. So here is the reciprocal Cid-B, which cannot bind to the A, inducing toxicity. So again, this is a chip away at 2x1. You don't need binding to get the phenotype. Here's that one, the Cid-B that cannot bind Cid-A, testing if Cid-A will rescue. If they bound, it would look like this. If they did not bind, you would see nothing, which is this. And then here, what they tried to do, this experiment worked for the Cin, but it's not working for the Cid system, which is they tried to flip both residues to match. And they're getting like, maybe they're getting like something with like that dot here. but I don't think they push it in the paper. They don't really talk about that. But it worked for the Cin system. Seemed like it didn't, that double flip, the double negation, seemed like it didn't work very well for the Cid system. The main take homes from the crystal structures, a couple interesting things, especially from my perspective. So one is where the position of this dub is. So to get crystal structures, often you have to optimize expression of the protein and optimize chunkifying the protein. And the only structures that they got crystallized were structures absent the dub. So when they look at these structures, right, like there's some connection, and here's where the dub would connect on. But the crystal structures themselves don't actually have the dub on there. So one thing that's interesting is a criticism that we have gotten for a long time has been I don't think they quite understand. So if you take Cid B and you add Cid A, and this is also true for Cin B plus Cin A, if you do these experiments, the Cin B that's complex with Cin A can still cut. And the Cid B that's complex with Cid A can still deubiquitinate. And so because we've been saying that CI is induced by toxic activity of the dub and the nuclease, people have been wrongly criticizing us, saying that initially they said these cannot possibly be the rescue factors because they're not inhibiting the enzymatic activity. They don't say that anymore. But people have always kind of not understood that. They have not understood, well, how can it be a rescue factor if you add the antidote and it's not inhibiting the B enzymatic activity? And what we have said in the eLife paper was what we noticed was that if you take Cid B and you look at all the proteins it's interacting with, and you take Cid B and you compare that to a complex of Cid B bound to Cid A and all the proteins it's interacting with, these are mutually exclusive sets. So every time Cid A is bound to Cid B, it's dramatically changing the interactions of the substrates that it's interacting with. They're mutually exclusive. And so our hypothesis for rescue, evidenced in the eLife paper, was that rescue isn't happening by inhibition of the catalytic site. It's happening by changing the interactions of what the B proteins are interacting with. And this crystal structure now in the interface and where the dub connects, this now really makes sense structurally because A is not interacting with the dub domain. Okay, so it makes sense that the dub domain can still sort of like cut, even though it's bound and complex to A. It still makes sense. It makes perfect sense. And so what we're arguing is that when this interface gets made, that's changing the interactions that B is looking for. So you might imagine that these interfaces that have now been discovered, these might be key substrate recognition motifs. And as soon as they're covered up by A, it's no longer grabbing onto the substrates that it needs to induce CI. So that makes perfect sense from what we published in the eLife paper. And I think this adds a little bit more detail. The other take home from the crystal structures is that the structural motifs of like what B is forming is very, very similar in the dub system as compared to the nuclease system. I mean, this looks pretty much identical to the nuclease domain, except it's got this dub domain, which would be tacked on as this little wart here on the bottom. So the key point here is that the Cin and the Cid interfaces are conserved, which again is evidence for that common ancestry, which we have been arguing all along, that these Cids, these Cins, these Cindies, they are a family of proteins that share common ancestry. I don't think anybody has debated that. Like nobody's been against that. They're clearly common ancestry. They're clearly like evolved from the same thing. And this dub thing just gets either tacked on or deleted in the Cin systems. Yeah, here's this is the exact point that I was making. The dub warhead of CID-B is not expected to be part of the interface because CID-A does not block its catalytic activity. Ooh, I like this. They use the nuclease warhead. I'm so happy to see this come out in published form. I'm so happy that Mark allowed that. That's unpredicted. Okay, here's an interesting point in the discussion worth highlighting and talking about. And it's a criticism that I've been making for a long time of some people who have alternative views in the field. They're taking these proteins, okay? They're taking these proteins with unknown functions, although they're not unknown functions anymore. And they're running them through these algorithms, right? Like interproscan and things like that, that are trying to detect essentially like homology to other proteins. And when they detect some homology to something, you'll get like a little fragment, okay? And so one of the arguments was that the CIDs and the A's have homology to this catalase rel domain. And then in the bioinformatic papers, what they say is that means that this gene might have this catalase function, and therefore you can't call it a dub system because there's these like other unknown things that might exist in the protein. My point all along has been a few different things. One, that argument is ad infinitum. You can always say, like, we don't understand the function of the N terminus, therefore you can't call it X. We don't understand the function of C terminus of A, therefore you can't call it X. Like, I can always do that. So it's a dumb argument. Dumb. The second thing is that Judy has this phrase that I've heard a lot from biochemists, which is like, some of these algorithms are junk in, junk out. Like, it's detecting a tiny bit of sequence. And just because it's like statistically significant, that doesn't mean that that domain actually has that function. It means it's got a little chunk that kind of looks like this one thing kind of over here. You cannot take these bioinformatic things without biochemical data and start claiming that they have these enzymatic activities, like without testing it. So I have never liked those because they're dumb arguments and they're not doing the biochemical tests. So they lack the biochemistry validation. Whereas what we have been saying all along is like, we found this dub domain and we tested it and we proved it. We found these nuclease domains and we tested them and we proved them. Therefore, like, it's never going to change. It's always going to have this dub activity, or in the case of CINs, it's always going to have this nuclease activity. Like, that's a proven fact. So like, that's one argument why the biochemical names are going to hold. And so here in this paragraph, what they're saying is they now look into that catalase related, the catalase rel domain. Okay, and they map it onto their crystal structure, and they say it only maps to this helix loop helix structure. And that structure in and of itself is completely insufficient to mediate the function of a catalase. So it's structural proof that, like, this is a bad idea. Like, don't just run these through distant homology algorithms, find these little chunkified things, and then take that as canon that that's the function of the protein. Like, do the biochemical work and prove it in a valid manner and demonstrate which residues are actually sufficient for that catalytic activity, and demonstrate that structurally it's conserved in a sense that it follows what's required to have that biochemical activity. They do the same thing with the PUF family RNA binding domain, which was predicted to be a feature of these systems. And it doesn't look like they are anymore. And there's a third one that was predicted, this STI-like transcription factor. They're saying, like, no, there isn't evidence for that. So the three things, right? Like, there was the, again, like, I don't want to harp on individual papers, but like, let's be more cautious. Urge caution. Let's not just like run these random homology things and say, oh, it's got this rel domain, it's got this puff domain, it's got this D-like transcription factor domain. Therefore, it has all of these activities. Therefore, we can't call it a dub. Let's not do that anymore because it's been proven to be a bad idea. What has been confirmed is the prediction of the nuclease domains, which was proven in the Chen paper and is now confirmed by the crystal structures. Very nice. It's interesting. Like when I read papers from, this is probably a bad bias, but when I read papers from kindred spirits, I can like once you start really learning the field, like people put in sentences that are soothing to our hypotheses. And then in other papers, when you read, they put in sentences that are pinpricks to our hypotheses. So reading some papers for me is very soothing, and reading other papers for me is very inflammatory. It's just an interesting point of science when you get so involved. Oh God. I won't say anything. I won't say anything about this, but you can pause the video, you can read this, and you can see what made me laugh. Very soothing. This is super interesting. The interfaces of CID-B are negatively charged, and the interfaces of CIN-B are positively charged. So they've essentially flipped, which is one reason, one reason arguing in this paper why there's a sort of like a bidirectional incompatibility between the CID and the CIN systems. Secondly, this is interesting because, again, we have been arguing that CID-B induces CI via the dub and CIN-B induces CI via the nuclease. The nuclease has to interact with DNA. And so it makes sense that these residues would be positively charged because the phosphate backbone of DNA is negatively charged. So that's going to be an interesting interaction. If that you could do a complex of CIN-B with DNA and see if you can find the interfaces of that. And if the interactions of CIN-B with the DNA when it's cutting are the same as the interactions of the residues, that is proof of rescue by sort of like inhibiting substrate functionality. That's really interesting. I should say that's indicative of rescue by inhibiting the substrate finding or the substrate interacting functionality. So here they're writing what I said in part one, which was that none of the TA model is not incongruent with a synchronization or a timing model. So this is well written. But it is incompatible with all models that are not claiming that rescue requires A interfacing with B. That's now a fact. That's now a fact. Nice. All right, very nice. Very nice.